Hello, this is Dr. Jabour, and today we're going to talk about ATP and enzymes. So ATP is a molecule, is a nucleotide that is going to be very important for cellular metabolism because it's the source of energy that the cell needs to perform a number of functions. ATP stands for adenosine triphosphate. I was looking for the second P, the first P. Adenosine triphosphate. So it's actually a ribonucleotide that contains the sugar ribose. Adenosine triphosphate contains the sugar ribose and the nitrogenous base adenine and 3 phosphate groups. So here's the structure, and then I'll tell you what my expectations are for remembering the structure. There's your ribose. So it's a 5-carbon sugar. Where are these carbons? They're here. 1, 2, 3, 4, and this one, 5. And this is an oxygen. So there's your ribose. And the hydroxyl groups are, either pointing down, or here it was pointing up before the attachment of the phosphates. But there's your ribose. At one end, you have 3 phosphate groups. Here's one, here's another one, and here's another one. What we normally write is just a P with a circle. And that's, for me, that's good enough. You don't need to memorize the actual structure here, even though it's pretty straightforward. It's a phosphate with 4 oxygens, and they're, all hooked together like that. But from now on, we just write it like this, these 3 phosphate groups. Like that. That's all we write. Okay. And they're attached to a ribose, the ribose sugar. So we can do this. There's your ribose sugar. And then there is a nucleotide. A nitrogenous base. And this nitrogenous base is adenine. We know that, for example, DNA has 4 different nitrogenous bases that makes up the DNA molecule. So ATP is using adenine. The other nitrogenous bases are so A, T, C, and G. Those are the 4 nitrogenous bases that you would find in the nucleotides forming DNA. So the nitrogenous base that is part of ATP is adenine. Okay, and for sure you don't have to memorize the structure of adenine. It's so we only write this. That's it. Okay, and really, what I normally write is just this A and then the 3 phosphates. This is my way of writing it. This is my ATP. Okay, so as long as you know that there are these 3 components to ATP, that's very important. So where's the focus going to be? It's going to be on the phosphate groups. It's the bonds, these covalent bonds, all covalent bonds in general, they hold energy. That's potential energy in the form of chemical energy, a covalent bond. And in this particular molecule, the covalent bonds that are going to be important are the ones that are linking the 3rd and the 2nd phosphate. And plucking off that 3rd phosphate group is going to release the energy that the cell needs for its various needs. So the bonds between the phosphate groups are very high in energy and can be broken by hydrolysis. This is a term you definitely need to know. Hydrolysis. It's a chemical reaction that breaks bonds by the addition of water. So H2O. So this water molecule is going to be split and used up to compensate for the phosphate that's going to be plucked off. So let me show you how that works. So this is the structure of ATP, okay? And in the hydrolysis chemical reaction, we have the water molecule that's going to, this is not showing the specifics. The water molecule is going to come in and basically going to, you know, it's going to be added to both phosphates that are going to get separated from each other. I just want to remember if it's shown later. It's not even shown later. Okay, so I'm not going to get into those details. Okay. So there is, let's say H and OH over here. So basically, because you're breaking off a bond between these two phosphate groups, now the phosphates are going to be missing a binding partner. Remember, each atom has to have a specific number of bonds with other atoms to fulfill the needs of the valence shell. This was covered in the basic chemistry lecture. So when you break a bond, you need to replace these lost connections with something. And that's where the water comes in. The water splits and becomes that new atom that these phosphates are going to be bound to. But this breakage of the bond releases all of this energy that is basically what we're expecting coming out of this bond. So breaking the second and third phosphate covalent bond releases so much energy. It's a great resource for the cell. It's unlike another kind of bond, it doesn't break a bond in the nitrogen space. It doesn't break a bond in a different molecule that's also floating in the cell. It's really ATP and that specific covalent bond between the second and third phosphate that just holds so much energy that when you break it, it basically creates like an explosion, which is why I kind of drew it like that. So that energy is basically what drives the cell to do the next thing that happens to require energy. For example, if we were in class together, I would ask you, give me an example of cellular activity that requires energy. And you would tell me, for example, a pump at the membrane. These proteins that are at the membrane that pump, for example, ions against their concentration gradient, that requires energy. So that energy is going to be used for pumping these ions against their gradient. So this is just one example of where ATP is needed by the cell. All right. Here's an example of how ATP can be used. So this is just another example. I gave the example of a pump at the membrane, but here's another example. So for example, if we need to transform glutamic acid into glutamine, which is an amino acid, it's a different amino acid, we are basically adding a nitrogen of an amine functional group. And this amine functional group has a positive delta G, because this is actually described as an endorganic reaction. So I've discussed endorganic and exergonic reactions in previous lectures. I'm going to, actually, I have it ready here just to kind of remind you. I had it ready open over here. So this is part of a different lecture. Chemical reactions that can be either exergonic because they're going to release energy, or endergonic as they are absorbing energy, for example, into a new covalent bond that's being created. And now this would be called an endorganic reaction. So endorganic reactions, I'm reminding you what we learned in a previous lecture, absorb free energy from the surroundings. So, hey, energy that can come out of an ATP molecule. And the reaction is usually not spontaneous, and that's why we need that energy from the ATP molecule. And that energy is then converted into this new bond and basically buried in there. I like to use the word buried because it's literally endo means in anyway, and it gets buried into that bond that, can be used, down the road if you break it. So back in that lecture, we had seen that the difference in the energy in the product versus the reactants was positive in an endergonic reaction, because you end up with more energy that got stored into those products than you had energy in the reactants to begin with. So the reactants don't have as much energy. They have to use the energy from an outside source like ATP, and then that energy gets buried into the new products. Okay. So a delta G that's positive reflects an endergonic reaction. So back to our, oops, that's another lecture. So back to this ATP and enzymes. Here the example is glutamic acid, which happens to be also an amino acid, can be transformed into glutamine by the addition of an amino group. And the addition of that amino group requires energy. So that's why you have a delta G that's positive over here. And that energy from, let's say, an ATP molecule is going to be buried into that new bond that got created over here. So here we go. So that's the energy in the form of ATP. That's your energy that's going to be used. And this is showing you the intermediate step where you have that third phosphate that got plucked off and basically used as a placeholder on glutamic acid. So this becomes a phosphorylated glutamic acid. So it's not your final product. And then the amino group arrives, and the amino group takes the place of that phosphate and becomes permanently attached to the original glutamic acid, and now it becomes glutamine. And so in the end, your ATP becomes an ADP molecule with the phosphate group being detached because it got used up as a placeholder here before the amino group arrived. So this ADP plus phosphate will end up being excuse me, I almost sneezed, will end up being regenerated, and ATP will be, reformed. And this is part of actually the discussion in the next lecture, which is cellular respiration, how the cell makes ATP. But right now we have ATP and we need to use it up. And this is the example of how we use up ATP. So speaking of energies, if we want to just kind of review the energy going on here, the energy to create glutamine is a delta G that's positive. So this is an endorgonic reaction. However, the energy to break ATP is an exergonic reaction with a delta G that's negative. So that's because the delta G, the energy in the reactant, which is ATP, is higher than in the product, which is ADP and phosphate. So that energy was inside that ATP, but ADP has less energy in it now because we broke a bond and we used up that energy. So that's why you see a delta G that's negative for the breakdown of ATP. But now, when you look at both, then your net delta G would end up being a negative delta G, only because that's how much energy is released from ATP. So that allows for that transfer of phosphate, the transfer of the amino group onto the glutamic acid to make it into a glutamine. All right. And here's the other example I had actually given you verbally even before I talked about the example of glutamic acid transformation. This is a transport protein. So I called it a pump. pumps at the membrane are proteins that will take solutes and bring them to the other side of the membrane where they are already concentrated. This is not a natural movement because naturally solutes will move down their concentration gradient, not up or against it. They're not going to move naturally into a compartment where they're already concentrated. That's not how it works. Normally, solutes will distribute across both compartments equally until it reaches equilibrium. That's the natural tendency of molecules. They're going to move to be equal on both sides of the membrane and reach equilibrium. So here we're moving them against their gradient. This is not natural, and it requires energy to kind of like push them, push them, push them. So this is a perfect example of where ATP is going to be used by the cell, right? So this is just two examples where the cell could use ATP, and there's thousands of places inside the cell where ATP is needed. Here's another example. Another example is the movement of vesicles across the cell. So this is really fascinating because the analogy I'm going to give you will show you how this is incredible how this works in the cell. So the vesicles inside the cells move from one place to another. And I don't have the lecture on cell organelles opened up here, but I want to remind you that we talked about organelles and vesicles inside of a cell, and vesicles are cell structures, little tiny membrane organelles that pop off the Golgi. And they pop off the Golgi, they contain something, and they will go towards the final destination of where that content is supposed to go. So if the content is, for example, collagen normally resides outside of the cell. The vesicle is going to go all the way to the edge of the cell and spill the collagen outside of the cell. If it's meant to deliver a protein somewhere else in the cell, for example, back into the nucleus or wherever that molecule is supposed to go, the vesicle is going to go towards that space. Now, these vesicles are not just floating aimlessly until one day maybe they'll reach their destination. They actually use rails inside the cell. And these rails are made of cytoskeletal proteins. So we also learned about cytoskeletal proteins back in that chapter of membrane organelles and the cytoskeleton. So the cytoskeleton, here I'll draw you a cell over here, and the cell has a nucleus and a bunch of, organelles like the rough ER, and then the Golgi, and then vesicles that can move through. Well, inside the cytoplasm, there's also cytoskeletal proteins. And cytoskeletal proteins are protein fibers that are just kind of floating inside the cytosol. Well, they have a job, and one of them act as rails. They're literally laying down in the cell just to be rails for vesicles to be moved from one place to another. Isn't that cool? And how do the vesicles move from one place to another is via motor proteins. And here's an example of a protein that is not destined to be outside the cell, not destined to be back into the nucleus, but destined to stay in the cytosol and to act as a motor protein on a cytoskeletal protein to help move vesicles from one place to another. Everybody has a job in the cell. So that movement, all this to say that the vesicles need energy to move from one place to another. So ATP used by vesicles to move, or actually used by the motor protein to move the vesicles down the cytoskeletal track, is an example of where ATP can be used. So ATP is a renewable energy source that can be regenerated by adding a phosphate group to ADP. So I had mentioned that. Normally it breaks down to release energy. That's really the goal here, is releasing the energy for the cell to use the energy for all of these things that we just talked about, but it's renewable. And energy from catabolism, exergonic reactions, and we'll see the examples in the next chapter. It's energy that's going to be used to basically bury it. It's taken and buried into the ATP molecule, and that way it's regenerated. Oops, I lost my picture here. Okay. Okay, so this was about ATP. I think you have a good idea of what ATP is and how ATP is very important for the cell, and a couple examples of where it can be used. So in the next chapter on cellular respiration, we'll talk about how is ATP actually produced constantly. So I showed you how it gets recycled, but where does it happen and what's the whole process of doing that? This is what will be the next chapter. So in this particular lecture, I want to talk about a second topic here. It's enzymes. Enzymes are important for chemical reactions, especially the ones that will happen very, very, very slowly without them. So even with a spontaneous reaction, meaning a reaction that's going to take place because it's just meant to go into that particular direction, and the reactants are going to combine and they're going to form a product. Sometimes these spontaneous reactions are not actually fast, and they're not fast enough for the cell. So enzymes are there to speed up that chemical reaction. And that's it. They're there to speed up the chemical reaction. So here's just one example of a chemical reaction, and there are thousands of chemical reactions that take place in the cell. Here's sucrose, that is actually a disaccharide that is going to, by hydrolysis, get split into a glucose and a fructose, because that's actually what sucrose is. It's made of glucose and fructose, 1 and 1. And they're bound together right here, and this forms sucrose. So when you split sucrose, you end up with a glucose and a fructose. And I want to show you where the water is used. This is actually a very clear example of hydrolysis. So let's put here the word hydrolysis, meaning we're splitting up this water molecule and we're using the atoms inside of that molecule to basically pluck in those holes. Let me do something different here. Right? If we break a bond here, then the oxygen here is going to be missing a linking partner, and the carbon is going to be missing a linking partner. So actually, in this drawing, it was this bond that was cut off. And you can see here, so we don't, instead of, to avoid having the carbon just, missing a bond, we give this carbon a hydroxyl group that we just plucked off of the water molecule. And we give this oxygen a hydrogen atom that is remaining from this water molecule. So now all the atoms are satisfied, right? This is back to the basic chemistry. All the atoms are satisfied with their, their bondings and the fulfillment of that valence shell. Remember, where it has to be full, and you end up with these two separate molecules. So how this happens is because there is this chemical reaction that takes place. It's spontaneous, but will take, I don't know, 1,000 years to happen. And I'm, of course, exaggerating, but it'll be so slow that it might never be actually visible. So enzymes speed up these types of reactions. So we describe them as being catalysts. They basically kind of like spark the reaction to take place. They speed up a reaction without being consumed by the reaction. So that's important too. These enzymes are just kind of players, and they're going to like create the spark and then watch everything happen. So in a different kind of lecture that goes in depth into how enzymes work, I have pictures that show how enzymes literally act as a couch, bringing the reactants together so the chemical reaction can actually take place. So here, this enzyme, which is called sucrase, by the way, I forgot to tell you that, and names that end with A's are usually enzymes. A polymerase, a ligase. These are words that you'll see in future lectures. And sucrase, all these are enzymes. Amylase is an enzyme in your saliva that breaks down carbohydrates. So anything with A's is usually an enzyme. So sucrase is going to act as a couch. It literally basically creates these two spots that are so comfy for sucrose and water molecules that they sit in there. It fits them perfect. These are like spaces that are perfect for the reactants. The enzyme is built for the reactants. So sucrase will only work with sucrose and water. It does not work with any other chemical reactions. So in these spots, you can see me, yes, you can see me. In these spots, the reactants sit in there, and it basically brings them close to each other, and now they can talk and say, hey, I need your hydroxyl group and I need your hydrogen because, I'm supposed to be splitting. Well, I'm here. Okay, great. And then they kind of, do their magic. If sucrase wasn't there, that couch bringing these two reactants together wouldn't exist. And, these reactants would be so far apart, they would never see each other, and it wouldn't happen. So that's what the enzyme does. It brings the reactants very close together so the reaction can spark and take place. So we need some free energy to do that. And as the bonds get created, we end up, burying that energy in those bonds. So the activation energy or free energy for activating this chemical reaction, it's the amount of energy that the reactants must absorb before chemical reaction will start. So you have to have some energy for this, everything to start up. So what do enzymes do? What is that related to our topic of enzymes, talking about enzymes? Well, what enzymes do, and that's the magic of enzymes, is they reduce the level of activation energy needed. So instead of needing so much energy to activate the chemical reaction, the energy needed for the chemical reaction to take place is so much lower. It becomes lower with the enzyme that with just a little bit of energy and poof, the reaction will take place. So this is what happens in the course of a reaction when there is an enzyme. When there's an enzyme that activation energy is reduced, so that way we don't need as much to get the reaction going. Okay, the energy that was in the reactant versus the energy that's now in the product, that doesn't change because that has nothing to do with the enzyme itself. That delta G is still going to be what it is. Okay, so here's the image that I was alluding to before as I was talking to my couch. This is the picture of an enzyme, a very specific enzyme, and enzymes are specific for specific substrates. So I've said this, 2 minutes ago, and I'm saying it again because that is a very important concept in this chapter. Enzymes are very specific to their substrates. Very specific. They literally hug the substrate in a glove way, where it's made specifically for that substrate. It will not be able to work with, galactose, or sucrase will only work with sucrose. It will not work with fructose or another kind of sugar. And the binding site is called the active site. This is the active site right in here. That's the binding site. The substrate binds in the active site in a very, very specific way. And it forms what we call an enzyme substrate complex. I mean, look, it's completely buried in there now. And the enzyme itself kind of closes up on the substrate and hugs it tight and basically says, usually brings two substrates together close. It says, okay, now you need to kind of like, get going and do the thing that you're supposed to be doing. So an example of sucrose, it was, and water, it was to like exchange bonds. So the active site, it's that pocket or groove on the surface of the enzyme where the catalysis is going to take place, and it's very specific to to the reactants. So not all substrates are going to fit in there. And I've been using the word substrate and reactants kind of interchangeably, because we also describe a reactant as a substrate. Yeah, okay, so I'm just kind of remembering here, reactant and substrate, right? So I'm now realizing for you, maybe that might have been confusing because there are two words that are basically being used for the same thing. Reactant is usually more of a word we see in chemistry, the reactant and the product. A substrate is usually more used in the context of biochemistry, where we're talking about an enzyme and its substrate. So now we're talking about an entire molecule in a cell. Okay. So the substrate is held in this active site by non-covalent bonds. So there's no like attachment on the enzyme. It just kind of gets hugged, just like a, like a hug, like that, without the actual, bonding. It's just a very tight fit, and the catalysis takes place. So the enzyme will convert the substrates into the products. So the substrates enter the active sites. And so here there's the illustration of two substrates. This picture only showed one substrate. But if we want to stick to our sucrose example, let's go back over here. There we go. We had two substrates. We had the sucrose and we had the water molecule. And these two substrates are going to kind of get very close to each other in the sucrase enzyme. So let's go back to this picture, right? So let's imagine this is sucrose, this is water. They both come in and fit perfectly in like puzzle pieces into that enzyme. So we call that the enzyme substrate complex. So they're held in an active site by very weak interactions, but they're still there, very close to each other. And this will be sufficient to literally, tweak at them. Substrates are converted to products. So this is where the bond within sucrose is broken. And now we have to kind of refill those open spots, and we use the water molecule to do that. So the products come out where it was glucose and fructose. I couldn't remember the second one. Glucose and fructose. So glucose and fructose come out. Now the enzyme is empty again, and it's available for another sucrose to be catalyzed into two monosaccharides. Okay, so this is just a reminder of how it's doing that. It's doing that by lowering the activation energy, so there's not as much energy needed to actually, conduct the catalysis. So how is that activation energy lowered? It's because of the scaffold where multiple substrates can come together in the appropriate orientation for a reaction to occur, basically for that very tight, specific puzzle pieces forming, right? So that's the scaffold. They're just kind of like fitting so perfect that they're like face to face, they're like, oh my gosh, you're here. Hi, how are you? And, sucrose and the water molecules are seeing each other like, okay, we're supposed to like exchange atoms, and I'm going to split, and then you're going to split, and then in the end we're going to have two monosaccharides. And this can only happen because of that scaffolding where both molecules are just like literally facing each other, like, hi. So the enzyme, in doing so, in bringing them together, it distorts the molecules, and it produces this, distorted transition state. And that's okay. That's part of the reaction. And in some cases, there's a microenvironment that's necessary for the catalysis. I can't think of a specific example at the top of my head right now, but it just makes it that the environment for these two substrates to be together is ideal in an enzyme. Okay. So this is a very a graph that's, if you get into biochemistry and, more and more advanced courses like that, this is something that you will see for sure. This is the enzyme activity. Here it's called velocity, means the speed of the enzyme, versus the substrate availability. So the more substrate we have, so this is more substrate. This is the concentration, right? When you see these brackets in chemistry, this means concentration. So the more substrate you have, the more enzyme velocity you have. So the enzyme is like, more substrates come in and chop, 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 chop. It's like taking them in and kind of like helping them get catalyzed. And then at some point, it's like kind of plateauing. It's plateauing. Because there's a maximum rate of catalysis that can be achieved, because eventually all enzymes' active sites will be filled. So you might have 10 enzymes and 1,000 substrates. Well, the 9,990 substrates that are left, they have to wait for their turn, because there are only 10 enzymes and only 10 substrates can be handled at a time. So I kind of exaggerated, but that's what that means. So it will really depend on the substrate concentration, and there will be a plateau where the enzyme can only do so much. So sometimes having too much of that substrate is useless because it's not going to get processed. So in a cell, enzymes degrade complex organic molecules into simple waste products, giving off energy in the process. So that's what the cell does with, the molecules that we have in our body. Sometimes they need to be broken down, and by doing so, it will release energy. So one way that energy can be released is by the transfer of electrons from one substance to another substance. So when we're talking about covalent bonds being broken, but then reconstituted in other molecules, right? Like the water molecule being kind of split, and you take OH and plug it into one molecule and take H and plug it into another molecule, you've split the covalent bond. But what does that actually mean? It means that the electrons that were in that covalent bond are now given to somebody else. So that's why we say the energy can be released the energy that's being released is done by the transfer of electrons from one substance to another substance. So that's what we're doing here. We're transferring these electrons from one substance to another. Yeah. So here's a little heads up on the next lecture, which is called cellular respiration. In the next lecture, we're going to see how ATP gets produced. So producing ATP is a big deal because the cell needs ATP. So we need a place to make ATP for the cell to be able to use it up. So ATP is made, and actually you can answer that question. If we were in a live class, I would ask you, hey, what do you remember about ATP and cells, and where is the site of ATP production in a cell? Dun, 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 dun. So this is what you're thinking. Okay, you can pause me and think a little bit, and now you're back. So which organelle is responsible for ATP production? We saw it was the mitochondria. Mitochondria, one mitochondrion, many mitochondria, that's the plural. Mitochondria in the cell are responsible for ATP production. So some cells that require a lot of ATP are very rich in mitochondria, will have more mitochondria than other cells. Some cells that have different job, that don't require that much energy, but have kind of a different role, will be rich in a different organelle that's just more important for the job of that particular cell. So every cell will have kind of like a different amount of each of these organelles. But for really a great example of a cell that requires a lot of energy is the sperm cell. The sperm cell that's deposited in the female reproductive tract for the purpose of reproduction has to swim all the way through the reproductive tract to meet the oocyte to fertilize. So it requires a lot of energy because that's a lot of movement, and sperm cells are rich in mitochondria because they need a lot of ATP. So what happens, all this to say that what happens in a mitochondrion is glycolysis and pyruvate processing and the citric acid cycle. All of this is called cellular respiration. Cellular respiration takes place in mitochondria and includes the process called glycolysis and includes the process called pyruvate processing and the process called citric acid cycle. So that's a lot of things going on, and that's why it's a separate chapter that's next. So here's just the big picture. Glycolysis is basically taking a glucose molecule, and I'm going to show you this because this is so important. A glucose molecule right here, and using the energy that we're going to get out of it to form ATP, 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 ATP. So glucose comes from the diet, the food you eat. The food you eat. So you need glucose. You need to fuel your body with some glucose. Your body needs glucose. Your brain cells use a lot of glucose. So glucose is going to be used up and broken down. And the process of breaking all these covalent bonds is basically taking electrons and putting them into intermediate molecules until we can form ATP. So ATP is really being produced along the way, but the most ATP is going to be done all the way at the end, after citric acid cycle. So in the next chapter, we'll see how we form 2 ATP during glycolysis. Here we see 2 ATP used up, but 2 ATP made. So this is not net. So it's actually going to be a little bit more precise in the next chapter. And here we see how we have even more ATP made here during the citric acid cycle. So what is this representing, this kind of stairway kind of thing? This is a change in free energy over time. So over time of this reaction of glycolysis and pyruvate and citric acid cycle, each of these drops across all of these reactions, energy is transferred to energy storing molecules. That includes ATP, but also NADH and FADH2. So these two molecules are going to be like your placeholder for these electrons until they can be used up at the end. So cellular respiration is the set of metabolic reactions that break down food molecules to produce energy in the form of ATP. It's brilliant. Food comes into our body, the cell is busy taking that food, breaking it down, and creating ATP molecules out of it for the body's own cells to survive. So here's glucose. And for the process to work, you need oxygen molecules. And the goal is to produce ATP. That's the goal. Now, the glucose requires oxygen. This is a must requirement. Must requirement. So glucose uses oxygen, and the breakdown of these two and recombination will produce a carbon dioxide and water molecules. So this is a reaction that I am going to ask you to memorize because it's really important in the concept of cellular respiration. So this is called an aerobic respiration because it requires oxygen. So make sure you understand that term, the term aerobic. This means in the presence of oxygen. You have to have it in the presence of oxygen. And in the end, cellular respiration allows you to produce 30 to 32 ATP molecules. That's a lot. So the three stages are glycolysis, pyruvate oxidation, and the citric acid cycle combined. That's the two. And then the third one, which is the first time I'm mentioning in here in this lecture, is oxidative phosphorylation. That's what it's called, and it's at the inner membrane of the mitochondrion. So these three stages are the focus of the next chapter that I'm going to get into. It's not an easy chapter, but I hope I'll explain it as clearly as possible. This is the big picture. You're going to have to know more than that, especially at the end of the next chapter. So again, glycolysis is first. This is what we do first. The cell, after glucose enters the cell, right? So you first have to have glucose in your diet. The glucose goes into the cells, and then the first thing that happens is glycolysis. So we end up with this intermediary molecule, pyruvate. We actually have two pyruvates. And then we need pyruvate oxidation. So you end up with acetyl-CoA, and acetyl-CoA is the molecule that's going to jump into that cycle that I've been talking about. And it's within that cycle that we're going to have NADH and FADH2 formed, and they will be used in the third phase of the cellular respiration. So this was the second phase. This is the third phase. It's called the oxidative phosphorylation. Oxidative phosphorylation. So there will be NADH2 and FADH used up. NADH and FADH2 used up in the electron transport process in the inner membrane of the mitochondrion. And this will drive something called chemiosmosis, and we'll end up with ATP. So I'm going to explain all of that in the next lecture on cellular respiration. Let me know if you have any questions. Thank you for watching this video.